DNA or RNA, what they're made up of are these individual nucleotides. We talked about that. The nucleotides, if we wanted to look at a prettier picture of the nucleotides, this is what we would see. So here's our nucleotide. So we have our carbohydrate here. In this particular case, deoxyribose, because this is a nucleotide of DNA. If this was an RNA molecule, this would be a ribose, regardless of whether it's ribose or deoxyribose. We have a nitrogen base included and a phosphate, just as it says in the notes. So, nucleotides are made up of a phosphate group, a monosaccharide, either deoxyribose or ribose, depending on whether it's DNA or RNA, and then a nitrogen base. So we have those three here, okay? So we've established what these particular nucleic acids are made up of. Just a string of these nucleotides. Again, just like an amino acid, string them together, you get a protein. No different here, it's just a different structure. And then we started to draw first DNA. DNA has two strands, right? We know that. So we have this double helix. So these two strands, they kind of twist. And the two strands themselves are once again made up of these individual nucleotides. So the circles are representing an individual nucleotide. Just stick them together and you get a strand. DNA, there's two strands. The two strands are connected together. Two pieces of a puzzle. Connect the two strands together. And we established what connects them together, right? What connects them together? These nitrogen bases. Okay? These four nitrogen bases are going to be connecting the two strands together, like two pieces of a puzzle. They're interwoven. Now, did we have? I don't remember exactly what I had as the bases. So that we're consistent, can somebody just rattle them off to me? What was the first one here on the left-hand side? C, CG, A, CA. All right. So we have nucleotides with those nitrogen bases on them, and we need to complement them on the other strand. Otherwise, the two strands are not going to connect with each other. So, what complements the C, as you can see in your notes, is G. So C and G are two particular molecules that fit together like two pieces of a puzzle. C, what's going to fit the A is the thymine. So on this side we'll have an A, here we have our G, here we have our T. So again, they interconnect. Now the two strands are connected to each other. What connects here to here? That's the phosphate. All right? That's what we call complementary base pairing. Those nitrogen bases complement each other. Two pieces of the puzzle. You will not see in a DNA molecule, if this is a C on this strand, that's not going to be a T. It just isn't going to happen. Or that's not going to be an A. Because the two pieces do not fit together. And so the strands would not connect with each other. So that's not the way it works. All right? So what do I want you to know? I want you to know complementary base pairing. So if I give you a number of nitrogen bases on the exam, you have to know what pairs with them. All right? What else do we have to know about DNA and nucleotides in general? And again, we're starting with DNA. So DNA, complementary base pairing, this is what we just drew in that picture. 46 DNA molecules in a nucleus. I think you probably already know that. I think you probably know that, yes. No. Okay? So we're not going to get into a lot of the details of 3', 5'. I'm not that interested in that. I'm concerned about that. Okay? Because then you'll have to flip-flop some things and it starts to cause some confusion. I'm not too worried about that. All right? We're just going to keep it to where I have it in the notes. An additional DNA molecule in the mitochondria. Now, when it comes to DNA, what's the job of DNA? Well, the DNA contains genetic information. Everybody's probably heard of genes, I would assume. That's what DNA is made up of. DNA is made up of a whole bunch of genes. So if we go back to our picture, here's a gene right there. I mean, it's a very short one because we don't have time to draw 50,000 literally nitrogen bases. That's a gene. The gene is GCT, that's the gene. A gene is dictated by the sequence of nitrogen bases. Here's another gene. That's a gene. AGT, that's a gene. There's 30,000 of them that make up a DNA molecule, all right? So our genetic makeup, 30,000 genes. Most of those genes are recipes for protein. Not all of them, most of them. All right, now I already mentioned to you that every single protein in your body is produced in your body. What produces protein in your body? Cells do. How do they make it? Well, they need a recipe first. Where do they get the recipe? DNA, the genes. That's the recipe for those particular proteins. And in a little bit, I'm going to show you how proteins are produced by taking this gene and doing stuff with it. All right? Transcription, translation, something that I'm sure most of you have heard of and have gone over. We're going to go over it again. All right? What else do I need you to know? Not only that genes are recipes for proteins, but I'm going to talk a little bit about this. Human Genome Project, before we get into some other things. Now, when it comes to the Human Genome Project, this is just a pretty picture of what we just drew. Again, here's DNA, it's two-stranded. How is this strand produced? How is this strand produced? A whole bunch of nucleotides. So let's take this right here, let's magnify it. This strand right here is this one. This strand right here is this one. Is this one right here. How are they put together? The bases. Or the nitrogen bases, I should say. TA, GC, AT, CG. We drew it. All right? Recipes for protein, that's what the genes are. We're going to see how it happens. Now, Human Genome Project. I'm not going to ask you exactly what it is. Who's heard of it? Yeah. Okay, that's actually more than what I usually see. All right, so you've heard of the Human Genome Project, and I'm not going to ask you, I'm not going to pick out anybody who knows what it is. I promise I won't pick up. I lied. Tell me what it is. No, I'm just kidding. I'm just kidding. Or you can if you want. Right. Yes, Ernest.
Well, the Human Genome Project is sort of one gene maps blood versus traits and attributes. In fact, when they first started the Human Genome Project, their initial hypothesis was as many as 100,000 genes. But in fact, there are even some studies that said that there are some genes for which are classified because it could indicate like fighting genes.
There's a lot of different kinds of genes within the genome itself. Yeah, and so not all of them have the same role. But again, most of the roles of genes are to be recipes for proteins. And so what the Human Genome Project is mapping out what all of the genes are in humans. Okay, up to that point, we didn't have that. And so that there were two people that did it, and it was actually done early. These two guys are the people that put it together. Francis Collins on the left was actually a professor at the University of Michigan. The guy on the right owned his own business. Okay, they're both scientists. They absolutely hated each other. All right, did not like each other at all. They were competing against each other to actually get the human genome mapped out. Government got them together, said, why don't you work together? And when they did, they got it actually done early. It was back in 1999. And it got a little blurb in the news. Okay, I think they might have made the cover of People magazine, and then that was pretty much it. Not a whole lot has been said about it. What's the big deal? Why am I spending a little bit of time on the Human Genome Project? I want to tell you the importance of the Human Genome Project. We know the genes of humans now. We know what they're supposed to look like. Now, everybody has a genetic footprint, but most of the genes that make up everybody in this room are conserved. There's just a little difference here and there. It makes you who you are. But most of it is all the same. So we know what the genes are supposed to look like. Now what? There's a lot of conditions out there. Most conditions, cancers, I talked about cystic fibrosis, I talked about muscular dystrophy, I talked about a lot of stuff that goes wrong in the body that has a genetic component to it. If we know what the genes are supposed to look like, and we have somebody with a particular condition, and we get their genetic makeup, and we look for anomalies within their genetic code, we're gonna know where those are supposed to be. So if this gene, for example, you have a person and you have a gene, and it's supposed to look like that, but this person's doesn't look like that. Now we know what the problem is. So what do? Let's cut out the bad gene and do what? Put a good gene in it. What did we just do? We just cured the disease. We just got rid of cystic fibrosis. We just got rid of muscular dystrophy. We just cured cancer. That's why this is so incredibly important, is that we can genetically engineer because we know what it's supposed to look like. And when it doesn't look like it's supposed to look, and we know that over here in this gene, this is the cystic fibrosis gene, right here. Let's take it out. Let's put another one in. Now, are there ethical issues here? We're playing God. Okay, obviously those are things that need to be discussed and already are being discussed when it comes to this type of technology. Are we there yet? No, we're not there yet. But we're going to be, at some point, going to be able to not only just genetically engineer things when it comes to curing diseases, but the baby's in the womb and it has a certain condition, you can genetically engineer it from that point as well. Okay, so there's all kinds of benefits to this human genome project, and nobody knows who the hell these guys are. Has anybody even heard of these guys? Seriously? I've never had more than one or two names. Okay, that's actually pretty darn good. These guys, when it comes to history, are going to be bigger than the Wright brothers. They're going to be bigger than the person who invented the printing press and the steam engine, all of it. Because what these two guys did and their groups potentially can pretty much wipe off every stinking disease on the planet right now. And nobody knows, I don't want to say nobody, but very few people know who the hell they are. And I don't know why. Because what they've done is incredibly important. Who knows now? Probably everybody. Okay? Now, I'm not saying anything bad about them. God bless them for making money, for doing absolutely nothing, or whatever the hell it is they do. Actually, they're geniuses for whatever they do. They should be studied, actually, for being able to market themselves the way that they do. But it seems that there's so many other things more important than important things. And again, I have nothing against the Kardashians. I will make fun of them from time to time, especially on the exams, but I have nothing against them whatsoever. Okay, but we still have to have some perspective when it comes to things, and hopefully at some point when the technologies are there to where these things are working the way that they think they're going to work, that these two get the attention that they deserve. So in the end, what do you need to know? The Human Genome Project is probably man's greatest accomplishment, or potentially it will be. That's pretty much what I want you to know from it. And I just wanted to mention it because it's going to get more attention eventually. It just has to. There's no other way around it. Okay? Anyway, that was a little tangent. Now let's move on. Now what? So we have our genes, the recipes for protein. How does the cell make the protein? That's the next thing that we're going to talk about here. That's when the RNA comes into place. There are 3 players involved: mRNA, rRNA, tRNA. And I'm going to show you what each of them does. So I told you that the genes themselves within the within the DNA molecule, that's the recipe. But in order to make the protein, we need to get the recipe in the cytoplasm. Where are these genes? They're in the nucleus, or they're in the mitochondrion. We're not making protein in the nucleus. The machinery is not there. So we need to get the mRNA, we need to get the gene, but not the gene, but we need to copy the gene. It's like if you're at my house and I have a recipe book, and you see this recipe that's in the book, and you can't stay at my house and make it, you gotta leave, and you ask, can I take your recipe book home with me? No, you can't have my recipe book. So what can you do instead? You can make a copy of my recipe, and you can take it to your house and make it. That's pretty much what's happening here. You can't get the DNA out of the nucleus. You can't remove the recipes. So a copy of the recipe needs to be made. That's what mRNA is. mRNA is a